This is an A-level biology presentation looking at epigenetics. To get started, using the words given to you on the screen, describe the structure of a chromosome. There are 1, 2, 3, 4, and 5 are all separate parts on sentences that you should be able to talk about, and you can go on and try the extension question, which is in terms of describe homologous pairs, diploid number, and epigenome. Pause the presentation, go through the best answer you can, and when you're ready you can restart the presentation. In terms of the structure, what you need to be able to talk about is we start with a molecule of DNA which is arranged into a double helix structure. We then need to talk about the fact that that double helix DNA associates with histones, and when it associates with 8 histones, it forms what is called a nucleosome, and that's what we see in number 3, those little cube structures. Those nucleosomes start to then form a coil, and they start to coil together, forming a substance called chromatin. The chromatin then starts to twist up into form loops, and it starts to twist up and condense even more to form chromosomes. We also mentioned an extension question in some terminology. Well, homologous pairs are pairs of chromosomes with the same genes, and a diploid number is the total number of chromosomes in an organism. So what is epigenetics? Well, up until now we've simply talked about DNA in terms of sets of nucleotides with bases on them, and we've talked about different processes such as transcription, translation, and DNA replication. Well, we're only touching the surface of the complexity of DNA molecules. Epigenetics actually refers to a set of markers that is found on DNA, and it basically results in genes being switched on and switched off. We can look at epigenetics in terms of different environmental factors and how the environmental factors can actually alter the DNA expression and how that altered DNA can actually be inherited. The epigenome, which is the part of the extension question earlier, refers to the markers that are actually found on the DNA or histone complexes and determine the chromatin shape. Remember, the chromatin is the structure that we get when we get the histones with the DNA wrapped around it starting to form into twisted coils. When we change the structure of the chromatin, we can use that to turn genes on and turn genes off. There are numerous examples of where epigenetic markers have actually been passed on between parent and offspring. What should happen is these epigenetic markers should be removed in gamete formation, but for some reason some of them slip through the net, and as a consequence certain characteristics are passed on. Now that doesn't sound like much, but you have to remember that these epigenetic markers can be changed by their environment, and there are examples of where environmental responses have essentially been genetically learned by organisms and then passed on to the next generation. The classic example is where mice were exposed to certain smells and electric shocks. As a consequence, they associated the electric shock with the smell. What would then happen is that those mice would be bred and their offspring would then be exposed to the same smell without any electric shock, without any negative impact. But yet the offspring showed exactly the same fear response as the adults did to the smell, even though they'd never experienced it before. And what was theorized is that the epigenetic markers that had turned on or turned off certain sections of DNA and certain genes in the parent to cause that fear response had been passed on to the offspring. At this point, we're going to start talking about specific epigenetic markers and the impact they can have on genes, transcription, and translation. What I'd like you to do is use the images in front of you to describe how methylation of DNA can switch genes off or silence genes. Pause the presentation, spend 2-3 minutes, try and work out what's going on, and when you're ready, restart the presentation. Well, what has happened is that there is a promoter site along the DNA which a transcription factor binds to, and that is the initial stages of transcription. If we stop that transcription factor binding with that promoter site, then we essentially stop transcription. As a consequence, when we methylate a promoter, it means that the transcription factor can no longer attach to that promoter. If it can attach to that promoter, then we will not start the process of transcription, and as a consequence, we've essentially turned the gene off. Methylation is associated with repeated sections of cytosine and guanine in the DNA. This is a slightly different way of representing the same thing. Spend a minute, try and interpret the diagram. When you're ready, you can restart the video. So pause now. Well, what's happening is we're talking about demethylation and methylation transferase. And in demethylation, we are taking the methyl groups away. So methylation transferase means that we're adding the methyl groups in. And when we add the methyl groups in, you can see the picture on the right hand side that each of the nucleosomes has basically packed together so that the chromatin has become much more condensed. As a consequence, it's not possible for the transcription factor to find the promoter site, and so transcription will not occur. We can also talk about acetylation, where we talk about acetyl groups or acetyl coenzyme A, but rather than attaching to the DNA, where the methyl groups attach to DNA, specifically the CG repeating sections of DNA, the acetyl groups, the acetyl coenzyme A, attaches to the histone, not the DNA. Have a look at the diagram and try and work out what the acetyl group is doing. Well, just like the methyl groups were able to condense the nucleosomes together and compact the chromatin so that it couldn't have a transcription factor binding to the promoter site, acetyl CoA works in a very similar way. What happens is that the acetyl group attaches to the histone and makes it more negative. As it is more negative, like charges repel, and as a consequence, it starts to make the nucleotide spread out as far as possible. If we do that, we are then exposing the promoter site and the transcription factor will attach. If we take the acetyl groups away, so deacetylation, we are taking away those negative charges, we are taking away that repelling force, and as a consequence, we have a compacting. And as a consequence, by deacetylation, we are going to switch or silence the gene because the chromatin has become condensed and the transcription factor cannot bind to the promoter site. So there are three main ways that methylation can lead to the development of cancer. Look at the pictures and try and work out what those three ways are. Pause the presentation. When you're ready to go through the answers, you can restart. Well, the three ways are that you could have something that should be switched on, a gene that should be switched on can be switched off. We can also have a problem in terms of a gene that should be switched off being switched on. And then the third type of issue that can arise from methylation and result in cancer is that we have certain genes that we'll look at later on, and those genes repair mutations that occur in DNA. If we methylate those sections, those genes are switched off. If we switch those genes off, then the mutations in the DNA are no longer being repaired, and as a consequence, you increase your risk of developing cancer cells. Well, now that we've got the three ways that we can have methylation lead to cancer, we also need to know about how epigenetics can be useful in medicine. Well, if we look at cancer, we know that methylation will turn genes off, so we can look for methylation sites and we can use drugs to actually target areas of heavy methylation. We can use other drugs that we can also switch off enzymes and destroy the enzymes, inhibit them, that would normally have methylation occurring. And we can also diagnose for looking for methylation and looking for the DNA for heavily methylated sites that we know are associated with people who are high risk for cancer. So the last thing we're going to look at is this whole concept of nature versus nurture. You can look at the diagram on the left hand side. There is additional guidance which I will show in a second, but you have where plants were grown and where plants were originally from, and then a description of what the plant looks like. The plants were taken from 3 different altitudes, just simply low, medium and high, and then they were replanted accordingly. And you've got a number of questions. Pause the presentation, have a go at question 1, 2, 3 and 4. If after a couple of minutes you're stuck and you want the additional guidance, you can restart the presentation to get the additional guidance, and then pause and restart whenever you're ready to go through your answers. So pause now, have an attempt. You can restart if you want to reveal the additional guidance. So the additional guidance to help you out is a simple 2 statements describing about the rows and the columns. Hopefully that will help you. If you've already had your best go at it and want to proceed straight on to the answers, just keep the presentation going. If you want to have another attempt at it, knowing that information you've just been given, pause the presentation now. And let's have a look at the answers. In question 1, you're asked, which factors have the biggest influence on the phenotype? Genetic or environmental? Well, in this particular case, it's the environment that is making the biggest difference, because it's the environment that we're changing and we're having a look at it in terms of how organisms are surviving. Question 2 says, what evidence is there to support your answer? Well, if only genetic, all the plants in the same column would be identical in phenotype. If it was only genetically influenced, then changing the environment would make no difference to the plant at all. So a plant from high altitude would look exactly the same no matter where you planted it. A medium altitude plant will look exactly the same no matter where you planted it. And a low altitude plant will look exactly the same regardless of where you planted it. And that's not the case, and that's how we know there's a genetic impact here. Question 3 is, what evidence is there to reject your answer? Well, the reason you can reject the answer is that some of the plants in the same column show the same phenotype when planted at different altitudes. So changing the environment is actually not changed the organism's appearance, which would tend to suggest that there is a genetic element in there. And then question 4, why is there more variation in the organisms from low altitude than from high altitude? And it comes down to the fact that if you have organisms that can survive in extreme environments and bring them into a less hostile environment, they will survive easily. If you take organisms that are only able to survive in less hostile environments and put them into a more hostile environment, they are not adapted to survive, and as a consequence will struggle and quite possibly die. There was an extension question looking about how you could conduct the same experiment with animals and humans, and the fact is that you would be very, very hard pressed to find any ethical grounds that you could justify this type of research with humans and expose them to different environments. Last thing, just to finish off, you have a number of terminologies in front of you. You should be able to give a definition of each one. Pause the presentation, go through your definitions, and when you're ready you can restart the presentation. So pause now. Well, here are the answers. Again, make sure you know these terminologies off by heart. Make sure that you are able to use them in sentences accurately. because these are the words that are going to get you the marks in the exam. In this lesson, we looked at what epigenetics was in terms of markers. We talked about the structure of DNA and histones. We talked about acetylation, and we talked about methylation and how they can be used as genes to be silenced or for genes to be switched on.